Okay, so I'm going to try to address 4 questions. So the fundamental techniques used to analyze resting state bold data are going to be important. How does a parcellation scheme, how can you make a parcellation out of your resting state bold data? The special challenges that result from the hierarchical organization of resting state networks. This is actually the really important point. And then I'm going to show what other people have done. You're comparing the resting state functional connectivity, derived parcellations, and how does that relate to other structures? So a question was asked, well, supposing you parcellate based on method X and method Y, should you get the same parcels? And the answer is, well, not necessarily. And the difference between those is maybe biologically interesting. So there are lots of ways to parcellate. And we mention here just anatomical. This is the AAL method. It's based on gyral anatomy. And I'm just going to tell you that many people who have compared the AAL parcellation to other ways of parcellation find that it does not perform very well. And probably the reason is that the brain folds on the basis of factors that have very little to do with neurobiologically important distinctions. In fact, even in twins, the brain doesn't fold the same way. So you shouldn't expect where a sulcus is and where a gyrus is to have a great deal of meaning, and indeed that is found when you use the AAL parcellation. This is originally what people meant when they want to divide the brain into pieces. You look at histology and you say, here's V1. V1 has the line of Gennari. The line of Gennari stops. This is the layer 4. And you go to V2, and V2 is different. And in this particular case, the break between the boundary between V1 and V2 is exceedingly sharply defined. You can do it in 2 different species. You can do it with different histological staining. But in this case, the boundary is very sharp. But Dr. Croxon showed you, if you go to prefrontal cortex, it's very difficult to see where one area begins, stops, and the other area begins. It's, in fact, rather uncertain. And this point about how boundaries are either sharp or diffuse or really hard to see at all is something I'm going to come back to over and over again. So this is Brodmann. This is 1908. And he did histological segmentation to create these sharp boundaries. Your first speaker showed you that Vogt did it in 1927, and when you compare this to Vogt, you don't see the same answer. So this is the first hint that a parcellation is maybe a slippery concept, because the assumption that you can always draw sharp boundaries turns out to be maybe not true. This is histology. It's a receptor staining. But again, this is Dr. Zillis's work. Between V1 and V2, you have different receptor densities, and this really augments your ability to see boundaries between region. You don't have to just stick to cell counting. You can make these receptor density maps and get huge amounts of very high dimensional information that makes one area of the brain look different from another area of the brain. Now, whether you wish to extend the concept of areal specialization to the notion of parcellation, which means sharp boundaries, that's yet another question. So there are, going now to imaging and talking about ways to parcellate the brain. This is from Dr. Glasser's paper. He'll probably show you the same slide. You can use imaging to do myelin mapping. And if you do that, you can see that some parts of the brain are very rich in myelin and others are very poor in myelin. And if you do task fMRI, now in contrast to what you just saw, here the task is done, the analysis is done by contrasting two tasks that are supposed to isolate a cognitive operation. So here the contrast is story versus math, and that makes a distinction between the, whatever is in a story different from a math problem isolates some kind of cognitive operation. So all of the mechanical stuff, the input, the output, all of that is subtracted away, and you just have the fine distinction between listening to a story or doing or listening to a math problem. So when you do that, you get this activation, which looks here like this. Now, if you do the functional connectivity with two different seeds, a seed here and a seed here. This seed is in the dorsal attention network, and it gives you connectivity with this part of the brain up here in the interparietal sulcus and down here towards the temporal lobe. And that functional connectivity is very different from what you put here, which is going to be part of a language-related network, which involves temporal lobe and Broca's area, and this piece of brain in the left hemisphere, which has to do with articulation. But I want to point out something, which is that, okay, you get these yellow spots. Where's my, yeah. So you get yellow blobs, and it looks like you've got some focality. But the reality is that you have all this red haze here that makes you wonder, what are we really talking about? If we really had parcels, how come we have all this red haze here instead of having just a yellow blob? Well, the brain isn't partitioned according to just simple parcels. Now, you can beat this problem by extracting high spatial frequency features, and you do that by looking for boundaries, basically spatial differentiation. If you spatially differentiate the myelin map, you get these very nice contours. If you spatially differentiate the task fMRI, you get, again, these very nice contours, and they do match, in this case, very nicely. And you can see that this piece sits right in the middle. I'm not sure I'm pointing the right. It's very hard to see. But the point is, by differentiating and looking at the strength of the gradient, you can make contours that form sharp boundaries, some places. One of the more, again, I point out, here you have sharp boundaries, but not sharp boundaries here. And in fact, if you ask, are there any parts of the brain where sharp boundaries completely surround a zone, the answer is very few. So this is some kind of clue about how the brain works. It has parcels, in quotes, but none of those parcels are surrounded by sharp functional connectivity gradients. Well, what does that mean? I don't know, but it's a question. Okay, so let's say you have a putative parcellation, which means sharp boundaries. Do they match task-evoked responses? Do they match histology? And by histology, I mean everything from cell counting to Zillus mapping to myelin mapping, structure. And then the question is, do parcels exhibit within parcel homogeneity? This is the sort of ground truth test which the functional connectivity parcellators tend to use. They say, if I have a real parcel, then the functional connectivity inside the parcel should be homogeneous. And so you can construct tests of how well that homogeneity holds up, and there's algebra for that. And you can rate the parcels for homogeneity and then say, well, this parcellation is better than that because the homogeneity within the parcels is better. This is a parcellation. This is a political map. But so here's France. So supposing you ask, well, supposing I look at function and I ask, where do people speak French? Well, they'll speak French here, but it turns out they also speak French in southern Switzerland, where we were last year. And, you know, it's not different. If you make a map about French speaking, it's going to look different than this. And here is Czechoslovakia. There are German-speaking people here. And this was of great consequence in 1938. So I want to say something about functional connectivity. What is it? Well, functional connectivity as we know it today with fMRI, there are antecedents going way, way back. But if you just take fMRI, this was discovered by Biswall in 1995. He did finger tapping bilaterally, got the task-evoked response, put a region where the response was maximal, extracted the bold time series from a resting state and then did correlation, and he got this map here, which is really remarkably similar, especially in the homotopic region here. It's interesting that this pre-SMA piece doesn't quite match. That's kind of interesting. But anyway, the idea is that if you do a task and you do resting state, by resting state correlation, you can very frequently see topographies which match what you get with the task. This is another example. I suppose it's important to point out that what you get depends on how you process the data. Here we do global signal regression. Global signal regression makes the distribution of correlations zero-centered, about zero-centered, so you get as many negatives as positives, and the topography isn't much changed, but I put this here because functional connectivity has been subjected to all sorts of insane critiques, and it's just a lot of nonsense. The point about functional connectivity is that if you put a seed here and you get this region here, you can get the same answer by putting a seed here and get more or less the same region. So the point is, if region A is connected to region B, then region B is connected to region A. So there's the reciprocal relation, which when you do matrices gives you a symmetric matrix, and symmetric matrices are always diagonalizable, and you get principal components. So, but you don't always have to do functional connectivity in the way that I just showed you. Two examples of seed-based functional connectivity. You can also do ICA. And this is a paper in which, well, this is Beckman's introduction to ICA, and you see that there are various topographies, including, interestingly, some negatives. But the point, this is just since this is an introduction to bold, I need to tell you that these are the two basic ways people do do bold functional connectivity. You can do seed, or you can do ICA. Spatial ICA is totally data-driven, whereas seed is directed towards a particular functional connectivity with the seed region. This maximizes component independence. This simply looks at the correlation map. This is very useful for eliminating noise. We should remember that ICA was first introduced as a way of denoising task fMRI, and it's quite effective for that. Whether you should use ICA to parcellate the brain is a very, very different question. This is an example of what happens when you do, this is a k-means result. So we talked about using k-means to take a matrix and turn it into parcels. And what was just shown is you select your number of clusters, and from the number of clusters, you have to tell k-means how many clusters. But supposing you don't tell it how many clusters, supposing you systematically examine how many clusters, you can sometimes see that there are better solutions and less good solutions. And this is a 7-cluster solution, and it more or less gives you functional connectivity resting state networks as have been found by other methods. But when you ask how does goodness of fit, or how does an error term in the parcellation depend on clusters, there's a peak here at 7, and there's a peak here around 17. And this has been obtained by more than one group. This is Yeo's. This is not k-means. It's a different way of dividing the symmetric matrix. But it's very similar algebraically. You get a 17 solution, and the 17 solution represents a minimum of badness. So the point is, how many clusters there are could be understood as the solution to a question of how well a clustering algorithm fits the data structure. Here's another way to divide the brain into functional pieces. If you do a graph theoretic measure of how well connections cluster together, this is information-based. This is an information-based criterion. It's not k-means, it's information. You just saw that you can do it more than one way. If you do that, then you get these resting state networks. I should point out that a resting state network solution of this type amounts to a parcellation. If the parcellation depends on exactly how you set up the clustering algorithm, in this case, the connection density, you have to wonder, what does that mean in terms of clusters being a fixed idea, or that there's only one solution, we simply have to find it. It may not be that there's only one solution. Here I'm showing you the 2-cluster solution obtained by ICA by the group in France. And this more or less corresponds to this is more or less the default mode network, and this is more or less the task positive network, as it has been called. You can further subdivide them, but this amounts to a sort of hierarchical clustering strategy based on ICA. So here's the 2-cluster solution. Here is the finer solution, and you can go finer and finer as you please. And when if you look at how these various ways of doing resting state network identification, how they compare to each other, they more or less do compare to each other reasonably well. So this tells us that by various methods, the brain can be divided into resting state networks, at least at the 7-cluster level, fairly confidently. Whether you can push it beyond 7 gets harder and harder, but the point is that you can do this. And for all practical purposes, this amounts to a segmentation, if not exactly a parcellation, because I keep saying a parcellation has to have boundaries, sharp boundaries around all patches. So you can do this with resting state fMRI using gradient mapping, or gradient intensity boundary mapping. So what's the idea? Here is my resting state matrix based on temporal correlation. This is simply the Pearson correlation obtained by computing the time series inner product over all voxel pairs in the brain. So I get a voxel by voxel matrix, and I can order the voxels so that they make some kind of sense in terms of resting state networks, dorsal attention, ventral attention, somatomotor, visual, and so on. So this is the 7-network solution. This is the functional connectivity matrix obtained from that way of looking at things. Now, supposing I do something else. Supposing instead of simply computing correlation, I say, I'm going to make a spatial map for every voxel. It's resting state correlation. Every voxel has a functional connectivity map, which is now a pattern in space. And I take all of those maps, which I get from all of my voxels, and I do a spatial inner product over all voxels. So I turn what was a correlation in time into a correlation over space. And if I do that, I get this very attractive matrix, which it has a much better contrast. And so with spatial correlation, I can get a much better segmentation of the brain. Once again, I just want to call your attention to the fact that, well, I'm going to skip that. So, I mean, the point is, if you do PCA, you get clusters, but those clusters aren't completely separate. Everything is overlapping. You just have to live with that. This is another illustration of the procedure I just said. It's not really different. It's just the same thing. So I'm going to move on. If you do this, and if you ask for moving the seed systematically across this boundary here, how the spatial inner product changes as I move across this boundary, you get very nice, sharp contours for change in functional connectivity going from one place to the next. And that gives you rise to these sharply demarcated, if you just get the magnitude of this gradient. So what is the slope here? What is the slope here? Where the slope is great, the contrast between where you were and where you're going is great, and it's the slope, which is the absolute value of the slope, which is quantified and put onto the brain surface. And when you do that, you get these nice sharp contours that illustrate where things change. But again, you do not have a full parcellation in which all the, quotes, parcels are surrounded by sharply demarcated boundaries. Again, this is some kind of secret about the brain which we don't understand yet. This is just another example of this. It's amusing to stare at these maps. Okay, so resting state gradients, resting state functional connectivity gradients, which is now based on spatial correlation between the two maps, are very unequal in magnitude, and the biological significance of this fact is not known. Okay, so supposing I have a hypothetical gradient, which, maybe I'll go back. How do you go back? Red, maybe? Yeah. Okay. So we take something like this, and we say, I'm going to ignore the fact that it's very bright here, but not particularly bright nearby. And I'm simply going to pump up the gradients until I get regions that are surrounded. I'm just going to take this and pay attention to weaker and weaker gradients until I can finally cut the brain up into something that is parcellated. And people do this now. This is the latest game in town. Having done that, you can ask, how good is my parcellation by computing the functional connectivity homogeneity within parcels? So when you do this, you have to compensate for the fact that it's a lot easier to be homogeneous if you're a small parcel than a large parcel. So some compensation for size needs to be made, and everybody does that, perhaps in slightly different ways. But the point is that, these are different parcellations, and they're supposed to show you that that they're different in homogeneity, but you can't see the bottom of the slide. Well, they're, okay, well.
They're in the same order as the top.
Oh, I see. Oh, okay, good. All right, so here's boundary mapping. Here is the, this is the information-based breaking up the matrix into parcels. This is somebody who has his own parcellation, somebody who has their parcellation. And if you ask, well, who's the most homogeneous, you can rate them this way. Of course, if you make a parcellation based on homogeneity, you'll look very good by this test. I'm actually going to show that as the latest, as at the end of the talk. This is very interesting. So supposing you do gradient-based parcellation around V1 and V2, you get this very nice boundary between V1 and V2. In other words, here is a case where, by doing the gradient map, you see something which reminds you of histology, and point out that the boundary between V1 and V2 happens to be the really strong histological boundary, no question about it. On the other hand, if you put a seed in if you do seed-based mapping, I don't see the seed here just because of my angle, but the way in which here's the seed. The functional connectivity does not look like this. It looks like this. In other words, it's strong here, it crosses the V1, V2 boundary more or less equally, and then moves weaker and weaker this way. So the seed-based map follows retinotopy, whereas the gradient-based map follows, let's just call it structure or histology. So this is a kind of an interesting contrast. Okay, supposing I want yet another way to break the brain up. I can train a pattern recognition machine to classify functional connectivity obtained from every voxel into a resting state network. And the machine was trained to classify into 7 networks. And so for every voxel in the brain, it will tell you how well the obtained functional connectivity map matches the archetypical functional connectivity associated with that network. And it's a perceptron, but the point is it gives you an answer. Every seed gives you an ideal, this is the ideal performance of the perceptron into mapping functional connectivity, or assigning network labels to each voxel-based functional connectivity map. If you apply it in individuals, it works rather well because it's a supervised classifier, not an unsupervised classifier like ICA. ICA is unsupervised. A trained classifier is supervised. In individuals, a supervised classifier works remarkably well, and I'm just showing the best and the median and the worst performer. And here is the winner-take-all map. You can take these. Okay, this is another important point. It's not true that when you ask, is any particular part of the brain a member of only one network? It's not true. Every part of the brain could be a member of more than one network. But if you ask which one is the strongest, you can always get an answer. And if you set things up that way, you always get a sharp answer. You always get a parcellation. There is always a boundary if you ask the question that way. And that's simply what's shown here. I want to draw your attention to this part of the brain here in the inferior frontal lobe, which has a sort of omega-like configuration. That's important because maybe that gyral anatomy makes it hard to compare across methodologies. That's going to come up later. This is just a group average of that winner-take-all parcellation. I'm showing to you because it's very attractive. But it does make a point. And the point is that the cerebellum, so here's the cerebellum result obtained from the cerebellum result obtained by running the classifier. The cerebellum was not included in the training set. So if I have here the language network which has left hemisphere specialization here in this articulation piece, and I check down here in the cerebellum, I see on the right side of the cerebellum language functional connectivity according to the classifier, even though it wasn't trained. So this is an example of generalization outside of the training set, which is an attractive feature of the classifier. And the point is, if you can do functional connectivity, if you can do the mapping in individuals, which you can when you have a supervised classifier, then you have results that are reliable enough to use clinically. And it is being used now clinically by neurosurgeons at our institution, so they know where not to cut. So now let's talk about cross-modal matching. If you have a classification based on resting state, how well does it match the task fMRI responses? So as was just said by the previous speaker, obviously the answer is going to depend on how you threshold the tasks. Because the reality is that if you do an activation with any task at all, you can, by lowering your statistical threshold, make response blobs grow and shrink as you please. But the notion that task fMRI and resting state fMRI do give you the same answer, this is not a parcellation in the sense of strict boundaries, but that they give you the same blobs, was, this is an often cited paper. It's actually not the first, but this is an often cited paper where you do ICA and you get this resting state network, and then you do task fMRI and you get something that looks very similar. I have to say that these resting state networks are rather localized, and I'm not surprised that the result looks as good as it does. But this is where the idea that resting state networks and tasks got first popularized, that they should match topographically, I mean. Here is something from our lab where you do the gradient-based boundary mapping, which is represented in blue, and you do a task, which is button push, and if you threshold your response appropriately, you can see that this orange stuff respects the blue boundaries, and you can do the same thing with reading, and you can do the same thing with a memory-related task. I don't think these results are misleading. I think it is true that if you do task fMRI, that the contours of the evoked responses more or less do match what you see with resting state. I don't think that's a wrong principle. I think it's a correct principle. So I want to point you to a very recent publication which really nicely articulates the issues here. If you want to know, has my resting state-based parcellation, how well does it perform in relation to task? And you want to see all of the ins and outs of what issues need to be addressed, what are the complications, what do I need to understand if I'm going to take this job on myself, then I think you could do worse than read this recent paper by Schaefer. It's Yeo's group, and it's now in bioRxiv, but it has just been accepted, I think, in Neuroimage. I'm not 100% it's Neuroimage, but I think it is Neuroimage. So this is interesting. There are 3 parcellations, which there are more than 3 that have been published, but they chose to compare 3. Here are the parcels from the Craddock group. Here is from Shen. Here is from their method, which their method is exceedingly analytically sophisticated. It starts with gradient-based. It includes in the optimization objective function a measure of homogeneity. So it's designed to maximize homogeneity. And once you do this, you also have to take into account distance, because since resting state networks are widely distributed, for example, the default mode network has pieces way in back and way in front, simply having a homogeneous answer or similar functional connectivity doesn't tell you that you're in a parcel. So you have to have a term that penalizes distance. Now, when you put all those terms together and you run the method, you get their parcellation, which is illustrated here. They call it, you know what, I can't even remember what this acronym stands for. But this is the Schaefer-Yeo parcellation paper, which is just out in bioRxiv. So yeah. Anyway, I encourage you to peruse these parcels or these boundaries, because the reality is they don't match. You can choose any part of the brain, and they just don't match. Now, why should they match? These boundaries are not well defined everywhere, but everybody has their favorite way of making up boundaries. Anyway, if you do that and ask how the latest set of boundaries relate to task activation, you can convince yourself that they do fairly well. Again, I don't think this is a misleading result. I think if you do task results, you're going to get responses which more or less match resting state. Here's what I was going to refer to before. If you do the now I'm blocking on what the red outline is. I think these are boundaries obtained by histology. You can see that they don't necessarily, you can get something that overflows, it doesn't match. In area 3, you can maybe get it to overflow. This is just a bar graph showing how their latest one does compared to the other parcellations. And again, your method works best using your test used to make your method. So the first question that was asked is, well, why should I do parcellation? And so an answer is, well, I need parcels so that I can compare groups. I want to evaluate something and I need parcels in which to evaluate it. Another is, I'm actually interested in parcellation per se as a measure of individual differences. And so I'm just pointing out, do I have any time left?
You're 5 minutes over.
Oh, well, I have to stop. Anyway, you can get individual differences if you do, this is from our group. So I'm finished.
All right. Thank you, Avi.